Welcome back to Caltech's 85th Seminar Day for our last but not least session number 4. Multum in Parvo, Phi-X, Multum in Parvo. Bill Clemens Jr. is the Arthur and Marianne Hanisch Memorial Professor in Biochemistry. He earned his PhD in 2000 from the University of Utah. He's also studied the structure of the bacterial ribosome at the Laboratory of Molecular Biology, I guess it would say laboratory, in Cambridge, UK, and conducted research at Harvard Medical School. Having joined Caltech's faculty in 2005, Dr. Clemens also chairs the President's Diversity Council. Welcome, Bill.
Thank you, Sandra.
Great.
All right, well,
take it away.
All right, my pleasure. Welcome to you all. It's actually a beautiful day here in Southern California, so I hope you all will get a chance to join us in person sometime soon. The title of my talk, Phi-X, Multum, and Parvo, is you'll understand it more in a minute why I think relevant to the story I'm going to tell you today. But really, ultimately, I'm going to tell you the story of the mechanism of protein antibiotic. And when we talk about antibiotics in this context, we're talking about killing bacteria. And as you understand, bacterial infections are an everyday problem that we've all encountered. Countering those has been a huge and important area of medical research, and so there's been a lot of time and effort spent in trying to discover new ways to actually kill bacteria. And so we're going to tell you about a new one, or a new way to think about one here today, about this protein antibiotic. The story itself really delves into the details of a phage called Phi-X, hence the title. And Multum in Parvo in Latin translates as much and little. And Phi-X, it's a bacteriophage. It's a tiny bacteriophage that does a lot of cool stuff. And so I think you'll agree with me by the end of this talk that's true. The other thing that's interesting is that because it's small, but it does a lot, right? And I think that's also, I love this title relative to Caltech, because Caltech also does a lot in a little. And if, like me, when you talk about your associations with Caltech and ask people to guess the scale, the numbers are always sort of 10 times the size of what our actual institution is. And I think that's, again, because Caltech is Multum and Parvo. Okay. I run a lab. I'm a biochemist, a structural biologist, and I have a team here that really does all the work. And so I don't want to take credit for the work that actually really came from these efforts. And the team really who brought this to fruition, the story I'm going to tell you about today, is here. Nadia Riera was a postdoc in the lab who was the first to actually make the breakthrough in the structural information that I'm going to share with you today. Karen Orta and Evelyn Lee are two grad students in the lab who are really pushing this through in the final stages. And so we're in the final processes of getting this paper out the door, and hopefully you'll agree that it's an exciting result. Okay, so this story really starts at Caltech. My connection to it starts at Caltech some time ago, roughly in the late 1950s. This is Bob Sinsheimer. Some of you will remember Bob. He was a faculty member here at Caltech from 1957 and 1977. During the last 10 years, he was actually chair of biology here. And Max Delbruck sort of famously created this virology group here at Caltech. And that really can be marked as sort of the birth of molecular biology in the way we think about it today, when a bunch of physicists were coming over to biology to actually develop these tools of biophysics to really aggressively paint a molecular picture of the biological phenomena that we were observing. And back in 1966, actually for Max Delbruck's 60th birthday, they decided to put together a book called Phage and the Origins of Molecular Biology, and asked a bunch of the sort of leaders in the field to write essays. And Bob Sinsheimer's book was essay was titled Phi-X, Multum, and Parvo. And so that's the title that I'm using here today. Now, I really like the sort of way he starts this, and so I'll just kind of let his words speak here. You know, he talks about why did he choose to work on Phi-X. And so Phi-X is this phage. It really started in a conversation in Max Delbruck's office, as many good scientific stories do, two people just talking about some problem in nature that they were interested in trying to understand. And in that conversation, roughly in the early 1950s, they basically began to think about the fact that Delbruck had really been leading these groups to use bacteriophage as a tool to actually understand what genes were, to understand genetic information and the transfer of genetic information. But they began to realize that many of the phages, which they refer to as T phages, were very complex, almost too complex for sort of simple analysis. And so they rationalized that actually finding smaller phages would actually be a benefit. And so that's what Sinsheimer set out to do. So he was actually at Iowa at the time when he started this work. And he identified two small phages, basically S13, S174. Turns out they're both just variations of the same phage. And because he thought these were good starts, he began to work on these and picked Phage 174 really to characterize his life's work in terms of research. And so we're going to talk about this today in some detail. But before we do that, I do want to sort of kind of preface sort of how the story started for me, because it actually started in a sort of similar way. And so this story started in a discussion with a close friend of mine, Tom Bernhardt, who is a professor at Harvard. Tom is a bacterial geneticist, and we were both Burroughs Wellcome Fellows. We'd actually previously been Damon Runyon Fellows together, and that's where our friendship started. We actually had been roommates as Damon Runyon Fellows. And so coming out of those relationships, at one point we were at the Burroughs Wellcome meeting in Dana Point, California, and we were sitting somewhere over here having a beer, and he told me this story of his PhD. And that really set me on this path that gets us to where we are today. And so that really interesting conversation got me excited, and it really centered on work that had come out of Ry Young's lab. And so Tom was a grad student with Ry Young at Texas A&M. And so you'll see and understand how the research coming out of Ry Young's group really informed everything that we've done in my group. All right, so I'm going to step back a little bit and paint the picture of sort of the pieces that you need to understand to be able to understand the story I'm going to tell you about today. So the two things to sort of take away from this slide are what a bilayer is, or a membrane, a biological membrane, and then the other is going to understand membranes and membrane proteins and membrane protein structure. And so we start with a membrane first. As you probably know, cells, you can think of a cell, a bacteria, a eukaryotic cell as sort of a balloon where there's a thin membrane that surrounds it and separates the inside from the outside. In biology, that's actually done by these lipid bilayers. Lipid bilayers are composed of molecules, and actually relatively small molecules relative to the scale of a membrane. And they look like this. And so these molecules, these lipids, as we refer to them, have polar head groups or hydrophilic head groups that like to actually interact with water. And then they have these long acyl chains, these sort of carbon chains that are hydrophobic, meaning that they don't want to be in water. Well, in fact, acyl chains don't care about whether they're in water or not, but water doesn't like them. And so water repels them. And so that repulsion from water forces them out of the water. And in doing so, causes them to have to create some new structure outside of water. And the structure based on the shape of these kinds of lipids that it can adopt is a lipid bilayer. And so what you can see here is just the copies of the lipid where they're sort of assembled together in a layer. There's a bilayer with sort of a reciprocal layer on the opposite side. And so that defines the membrane, which would be right here, would be the middle of the membrane. But this is going to go off in all directions, right? And so you can think of this as a two-dimensional layer that goes off in every direction. The properties of this are that the central part is inaccessible to water. Water does not want to go in there, and it won't. So the effects of that are that these forces drive these things together to actually form these actually fairly strong interactions that prevent things from crossing it. Now that's important, right? Life requires the fact that you have the biochemistry of the inside of a cell separated from the environmental chemistry of the outside of the cell. And so membrane bilayers are what do that, and very early in evolution, these had to evolve to do this process. Now here, what I'm showing in this image here, and it's a movie, and we're going to run it in a second. This is the structure of a membrane protein TAT-C that my group had solved some years ago. I want to highlight a few things. One, with membrane proteins, we often describe them as having what we call transmembrane domains. And so these cylinders represent these transmembrane domains, which are alpha helices. It's a structural feature of proteins, first discovered actually by Linus Pauling here at Caltech. Here, these transmembrane domains are these alpha helices that cross the bilayer. Now, it's important to remember that proteins are all just polymers, and so we're just following this polymer chain, and you can actually trace the whole polymer throughout this. And so it's a single polymer that's assembled together that contains an amino acid polymer. By its nature, amino acids are polar, and so this polar polymer has to somehow fold and assemble in this hydrophobic environment, because membrane proteins exist in this lipid bilayer. They do this because they expose very hydrophobic segments to the surface here. And so my lab is actually really interested in understanding that sort of broadly and some of the processes that are engaged in that. But in this context, what I also want you to take home is the fact that we're looking at a bilayer. So computationally, what we've done is taken this lipid bilayer, embedded our protein in it, and then now we're just going to run a molecular dynamics simulation where computationally we're just going to let the atoms do what atoms do. And red are our water molecules, and these red dots are water molecules, and this is the bilayer. And when we let this movie go, what we're going to see basically is that everything's in motion. These bilayers, because these are single molecules, are actually free to move relative to each other. And so they do move relative to each other. And so this is really a dynamic, fluid environment that these proteins have to exist in. And so this is just an important sort of way to think about membrane bilayers. Now we're going to put this bilayer in context, right? So we already talked about the fact that the bacteria is sort of a balloon that has surrounded by a bilayer. But we can look at that in a little more detail. And this is actually artwork from David Goodsell down at Scripps. He's a professor who does who really has a sort of beautiful way of capturing biological understanding at molecular detail using watercolors. And so this is a watercolor of E. coli. The dimensions of this, an E. coli cell from front to back is about 2 microns, right? So if you take a millimeter divided 1,000 times, you're at the scale of what a bacteria looks like. And here you can see that this is surrounded in green in this coloring of a membrane that defines the outside and the inside. On the inside is what we call the cytoplasm, right? That defines the inside. And in orange here is all the DNA that encodes all the information for the bacteria to be able to go through its full life cycle. We can zoom in here, and so in fact he has another picture that really zooms in on this and sort of shows what we looks like when we zoom in, and we can see all kinds of features here. And they're mostly, these aren't important except to just highlight the fact that here we have the cytoplasm defined on the inside where all the sort of biochemistry is happening. And there's a cell wall which represents this layer in green here that defines the inside and the outside. One feature to note is this E. coli. E. coli, if you recall, is a gram-negative bacteria. Gram-negative bacteria really are defined by the fact that they have 2 membranes, 2 biological membranes that basically surround the cell. And between those biological membranes is what we call the periplasm. And so cytoplasm, periplasm, and then outside the cell. And the periplasm is where this peptoglycan layer resides, which we're going to be talking a lot about today. Bacterial cell walls is just another present bacterial cell walls. And it's just from a textbook called Essentials of Glycobiology. And here we're representing a gram-negative cell. We have a cytoplasmic membrane, an outer membrane, and the periplasm here. In this type of figure, all these geometric shapes here, squares and circles, are all sugars. Right? They're all sugar molecules, and they all form different kinds of polymers and different kinds of structures that are essential to the stability and the maintenance of the bacterial cell wall. The most exciting of these sugar polymers for our interest today are the peptoglycan layer. And so here we actually have long polymers, we call these long polymers of sugar, polysaccharides, where we have 2 different repeating sugars that form these long strands, and they're actually cross-linked by amino acids or a peptide. And so the name peptoglycan really just derives from the fact that it contains peptides and sugars. A peptoglycan completely surrounds the entire cell, and in fact this is true for gram-positive bacteria as well. And the role of peptoglycan is to provide strength, and so this is actually a meshwork of polymers that's completely covalently attached, and so it's a single molecule that surrounds the whole cell. And in fact, it's responsible, I think about it as chainmail, because it's responsible for preventing any sort of pressures that can come onto the cell from the inside or the outside that would lead to the cell to rupture. And so it's important to have that chainmail that surrounds it. And in fact, this molecule is so robust that if you were to take this bacterial cell, and here what they've done is he's taken this bacteria, Aquaspirillum serpens, boiled it in SDS, so a very strong detergent, boiled it, and then spun it down, and what you get out of that is a single molecule of peptoglycan. And the thing you'll note sort of right away is actually it completely maintains the shape of the molecule. And that's just like if you take the chainmail off, the chainmail still looks like it looked on you, right? It doesn't deform because it's responsible for providing this sort of exoskeleton, if you will, of the bacteria. And so that means it's important. The other thing that's interesting about peptidoglycan is it's unique to bacteria. And so when you say something's important and essential and unique to bacteria, this becomes a really important target for the development of small molecules that can inhibit the biosynthesis of this and perhaps then allow you to develop new antibiotics. All right, so going back to the early days in the sort of 60s, they mapped out all the different players and how we build up a peptidoglycan. And so I'm just going to walk you through this kind of quickly here. Here we're going to see our cytoplasmic membrane in gray here. On the cytoplasmic side, in the cytoplasm, we're going to start with actually a soluble molecule. And this soluble molecule is this uridine diphosphate sugar. That sugar is just a modified glucose, which we call N-acetylglucosamine or a GLICNAC. And so there's the uridine diphosphoglycnac. That's going to get modified to actually modify this to have a new type of sugar, which we call a murenic acid or a murinac. And so this UDP, uracil diphosphomurinac, gets modified where a series of enzymes are then going to add amino acids to it. And they're going to basically end up with 5 amino acids attached to this murenic acid. And so this now we call this uracil or uridine diphosphomurinac pentapeptide. This is the soluble component. This is really all of these steps are essential to this process. The first membrane active step for this is a transfer of this phospho sugar pentapeptide onto a phospholipid. This lipid we call bactoprenyl or undecaprenyl phosphate, where it's got a single phosphate group onto this. This phosphate actually provides the energy for the transfer, so it's sort of a high energy reaction where you get this phospho sugar transferred onto this phospholipid to generate what we refer to as lipid 1, right? There's a lipid molecule, 2 phosphates, and the sugar pentapeptide. That's catalyzed by an enzyme MRAY, and MRAY is going to be one of the stars of the show today. And this is actually a really broad area of research in my group. After MRAY has generated lipid 1, another sugar comes in, another GLICNAC comes in and gets transferred onto this to make a disaccharide, so these 2 sugars with a pentapeptide, which we refer to as lipid 2. This is all done in the cytoplasm, but now it's on the wrong side of the membrane because the action for making peptidoglycan all happens in the periplasm. And so MURJ is an enzyme that catalyzes the transfer of this soluble components across the membrane into the periplasm, where then a series of enzymes can work together to build this sort of simple building block into this polymer of sugar that's cross-linked by amino acids. So I've already said that MRAY is the star of the show. And in fact, if we look at the reactions that it catalyzes this really sort of interesting reaction where we're going to take this molecule, which we refer to as Park's nucleotide, which is the soluble component. It's got the diphosphate, and this phosphate sugar pentapeptide. So the phosphate is going to be transferred onto this phosphate of this undecaprenyl phosphate or bactoprenyl. This has 55 carbons in it, right? So it's an extremely long acyl chain that exists in the bilayer, and the phosphate is going to get transferred onto it here to generate this kind of high energy linkage that's critical for making peptidoglycan. All right, and so that's basically how peptidoglycan is made. Now, as already hinted at, this is a great target for antibiotics, and in fact, the most famous of antibiotics that everyone's familiar with is penicillin. Penicillin is a part of a family of antibiotics called beta-lactams. And upon the discovery of penicillin, and actually more importantly, the abilities by chemists to generate a way to synthesize penicillin at industrial scales allowed penicillin to really take over the world as sort of a major drug. Now, the surprise is that pretty quickly, people thought you had this miracle drug, but then pretty quickly bacteria started developing resistance to it. And this is going to be the constant problem that we're going to see with small molecule antibiotics. But penicillin was the first. And after that, obviously, lots of things have come along. And in fact, as a father, I basically learned a lot about in dealing with ear infections and things like this, that beta-lactams are still actually a major class of antibiotics that we use prescriptively every day. But there's others as well, and I'm just going to point out that there's a couple of different places, like bacitracin, one probably most folks have heard of. And these are all different points in the pathway that can be inhibited that have been shown to be able to kill bacteria in a clinical setting. Most notable here are the vancomycin and minomycin, which also basically inhibit the stitching process for peptidoglycan. Vancomycin and minomycin are kind of well known as drugs of last resort. And so if you actually have an infection that nothing, you often end up getting given intravenously minomycin or vancomycin to actually hopefully cure your infection. The problem is that there are actually now lots of bacteria that have developed resistance to vancomycin and minomycin. And so we're actually, in some cases, we have no drugs to treat some infections. And so the question is, so how do we actually come up with new drugs? Ways to do that are actually to kind of mimic nature a little bit. And one way to mimic nature is to identify small molecules that nature's already generated to inhibit the pathway. Two of interest to my group are ones that inhibit MRAY and MUR-G, which is this is enzyme here. MRAY, basically, tunicamycin is a long-known antibiotic that some of you may have heard of before. But there's a whole class of small molecules that will inhibit this. And these are all natural products discovered through natural product discovery that inhibit these various these various enzymes. And so in collaboration with a synthetic chemist, we've been working to try and actually leverage these to develop new drugs. And I won't have time to go into that today, but this is sort of an exciting area of research. The real focus for today is going to be on another way that nature's evolved things to actually be able to inhibit this pathway. And the way it's done this is actually phages. So Bacteriophage have actually developed tools to actually inhibit Peptoglycan biosynthesis. And so this is a theme we're going to come back to over and over again. But examples are the Q beta and the M phage, and then Phi-X. And so Phi-X inhibits MRAY. So MRAY, again, as I said, is one of the stars of the show. The structure of MRAY was solved some years ago by the Lee lab at Duke. In the Lee lab, what they saw was that MRAY, they actually solved it from a structure from a bacteria called Aquifex aeolicus. This is a bacteria that grows at about 95 degrees Celsius. And so it's a really sort of wild bacteria. But we'll see some features that seem to be true for all MRAYs. And so here, this is what I'm showing you now. There's two molecules of MRAY. So MRAY exists as a dimer. There's two copies that basically have are split down here in the middle, and you basically have rotational symmetry. So if you take a molecule here and you rotate it around, you get the other molecule. This is how they exist in the membrane. And the cytoplasm will always be on the bottom, the periplasm here on top. And if you look at the structure here, we're going to focus on one monomer in this case. In this monomer, you can see there's sort of interesting features. So there's one helix here. This helix has this really strong kink in it, and then this steep angle in the plane of the membrane, which is actually a pretty surprising feature and a very unusual feature of MRAY. This defines basically one side of a groove that's formed by this helix that sits back in this angled helix here. And in fact, as we'll see numerous times, the active site really is defined by this groove here. And so there's two active sites in this case, and they're both on opposite sides with the way that MRAY interacts. We can look at MRAY in more detail, and so this is just a video, which I hope works for everyone. What we can see in the video here is that we're now just going to circle around one copy of MRAY, and again, there's this sort of angled helices. And what you can see then at the back of this is at the back of that groove that's formed is where the active site is, right? So that's where that phosphate is going to be transferred on the other phosphate to generate that lipid-1 molecule. You can see this a little bit here, hopefully, in this sort of surface representation of the protein. Remember, proteins are actually three-dimensional objects, and even though we cartoon them here, you can see that they actually have surface features, and there's a groove that's formed back here where that small molecule active site is localized. All right, so my group's been interested in this for a long time, and in fact, we still don't really understand exactly how the mechanism of this happens. This probably looks a little intimidating, so I'm not going to go through the data here, except to say that we've begun to look at residues that we know are highly conserved, meaning that they're found in all copies of MRAY, suggesting that they're important functionally. And what I want to draw your eyes to here is that we've got, if we look at this binding pocket, right, this is our angled helix. It actually has this sort of extended loop that connects to it. And in that loop, there are some conserved residues. These are actually histidine residues, but these are completely conserved in this family of enzymes. And so we know they're important catalytically. On the opposite side, these helices here and these other helices really define a pocket where the uridine diphosphate is going to be recognized. And these are all critically important too. And so we've done assays to really assay the effects of mutations here on both activity and binding in this. But just to really convince you then that the active site's going to lie here, and it's going to encompass pieces all around the membrane here. All right, so we're going to change gears, and we're going to go back to phage. So bacteriophage gets this from the Latin for eater, right? So phage is Latin to eat. And bacteriophage is really interesting. Some of you may have never thought of bacteriophage before, but if you actually think about bacteriophage, what we discover is that is that if you do some estimates, this is just back of the envelope calculations actually that were done by Rob Phillips here at Caltech. And I just found these fascinating, so I include this here for you. The estimate is there's about 10 to the 30th particles of phage, right? That's a crazy number. It's sort of hard to get your head around. But if each one of those phages are on average about 150 megadaltons, if you calculate the total biomass of phage on the planet, you get about 10 to the 11th kilograms. And at 10 to the 11th kilograms, basically compare that to the total mass of humans on the planet, about 10 to the 8th kilograms. That means that phage are 3 orders of magnitude larger biomass on the planet. And so really it's a phage planet, and all the rest of us are just players in it. All right, so phage, just from a purely interesting perspective from understanding the biology of phage, is really an important problem. But let's focus more on sort of how we can use phage. And so phage was first discovered by, first observed by Frederick Twort in a paper he published in 1915, where he first observed what he thought were these and in fact are these tiny little viruses that seemed to be involved in targeting bacteria, and this is done in England. Probably the most important player in this whole story, though, is Félix de Herelle. So Félix de Herelle was a French-Canadian working in Paris. And he basically described this invisible microbe that could kill the bacteria responsible for dysentery. He published this in 1917, around the same time. And what was really amazing about that work is that Félix de Herelle immediately realized the importance of phage and the ways that you could use it. And so, why study phage at all? Well, it turns out that phage actually may hold the promise of solving our multi-drug resistant bacteria problem. And in fact, this was first evidenced by Félix de Herelle. So when he made this observation in 1917, he began to realize that he could actually kill bacteria using these viruses. And he thought, well, we have these issues with these bacterial infections, dysentery being a major problem in Paris at the time. And so, so he went on to actually purify, or at least make relatively pure cocktails of phage, which he then first drank himself and then gave to some of his assistants and showed that it was perfectly safe to consume as a person. And then he basically took that and gave it to a 12-year-old child who was in a really bad state with dysentery. And giving this phage cocktail to this child actually immediately cured him. Within a few days, the child was back up and running. And so this was the first actual evidence, or the first clinical use of a phage. But as we'll see, this is actually now really an important area of research for the world. So de Herelle founded this laboratory du Bacteriophage in Paris. And this is just examples of some samples. These are collected phage from all kinds of environments. Most of the places you find phage are where you find the bacteria. And so much of this is collected from sewers, for example, although you can get them from fecal samples as well. This is an example of sort of a product that would come out of the lab of these bacteriophage and these ampules that could be given to people. And so really they started this effort to try and use phage as a therapy. The problem is that phage therapy basically began to take hold at the same time that penicillin was discovered and the synthesis of penicillin happened. And so pretty rapidly the pharmaceutical companies, at least what became pharmaceutical companies, I guess, appreciated how powerful penicillin was going to be and just began to market it really effectively. And so penicillin took over, and phage kind of got lost as an effort for therapies in the West. But actually, folks who had studied at this bacteriophage laboratory in Paris went on to actually found other areas of studies. And in fact, even though this had fallen away in the West, in the Soviet Union, this was an active area of research for many years. And many people were cured with bacterial infections by phage in the Soviet Union out of a very famous research lab in Georgia. And so phage therapy never went away, but it was really in the background for sort of much of research efforts to target bacteria. But that's changed. And so this is actually a really interesting story. So this book, The Perfect Predator, written by Stephanie Strathdy and her husband, Thomas Patterson. And she's an epidemiologist and was actually studying HIV. And so this was her area from an epidemiology perspective, her area of work. But her and her husband were actually on a vacation in Egypt, and he developed this infection from Acinetobacter baumannii. It turns out that this was actually a completely multi-drug resistant bacteria, and so no known therapies were able to stop this infection. He was dying, and there was no way to save him. And so Dr. Strathy basically turned to Phage, and she just did her homework on the web and discovered that there was this possibility of doing this. And she contacted people to get help. And one of those people she contacted turned out to be Ry Young, who was actually a significant player in the story. So of course, they were able to create a phage cocktail to save his life, and so they now have a center down in UCSD for the Center for Innovative Phage Applications and Therapeutics. But really interestingly is the fact that that this is now kind of exploded, and so phage lots of small companies have been started trying to develop phages, both therapeutics for people, also for animals, also for treating. For example, we can basically imagine, if you've got a crop of lettuce that you're bringing in that you're concerned may have a E. coli infection, if you could wash it with phage, kill off all the E. coli, and then send it off, that could be a real solution to some of those problems as well. And so there's a lot of really exciting research being done on phage. And so it's important then to try and understand phage better. Bacteriophage itself looks something like this. This is probably how you picture it. There's the head group. Within the head group, there's this DNA. There's this long neck that performs a sort of injection needle. When the bacteriophage finds its bacterial host, it shoots that DNA into the bacteria. What do you get from that? You get something that looks like this. So this bacteria injects its genome into this. It takes over the host and then makes more copies of itself, eventually assembling new particles. And then in the final step, it has to release from the cell. And a key component of that is it has to break out of the peptoglycan layer, because the peptoglycan layer, again, is this chainmail that if it can't break through, it will be stuck. A standard T phage like T4 is just exemplified here. So this is a T4 phage. It's got this massive genome, reasonably massive genome, about 170 kilobase pairs, about 300 genes that are encoded, that are all required to assemble this particle. So it has to assemble this particle, identify a new host, inject itself, take over the host, make new particles, and release. And in the release phase, what it does, what you see at the end of that release phase, is this sort of bursting out of the bacteria. So this is an EM image showing this bursting of these phages. And the result then is that you now have new phage particles that can go off. And in fact, lots of other bacteria get this rapid amplification of killing. They do this using two components. One is a protein called a holin, which is a membrane protein that at certain concentrations will punch holes in the membrane. That will kill the bacteria, but it's not sufficient to actually release, because you have to break out of the peptoglycan. And so it does this by releasing enzymes called endolysins or lysozymes that will release, they chew up the peptoglycan, and then you can escape. All right, so Phaex. So how do we get back to Phaex? And so the first paper that described Phaex was actually a paper published by these two folks. At the time, Vladimir Sertek was actually running the laboratory in Paris, and Bulgakov was one of his assistants. And they have this paper where they actually went through and categorized all these phages that they were identifying. And what's sort of cool is that in this categorization, they actually going through and saying, here's category 1, 2, 3, 4, 5, 6. And Phaex was the 10th category that they had identified. And this was the 174th experiment that they had done in this context. And so they just called it Phaex 174, Phi meaning phage. And so the name really has no other interesting reason except for that. All right, so this was basically discovered for the first time, characterized, or at least named in 1935. And then we're going to scroll forward, right? So Sinsheimer arrived at Caltech in 1957 and began working on Phaex. And this paper came out in 1966 from Clyde Hutchison, who was a grad student in Sinsheimer's lab. And Hutchison basically published this paper that describes how the first example of how the lysis gene works. All right, so Phaex has a lot of historical firsts. It was the first, it was actually in that first phage cocktail that was given. And so Phaex goes back to the earliest days of phage. And in fact, it's everywhere. Lots of other firsts, first single-stranded DNA, first circular DNA, first self-replicating DNA. To the probably the most interesting thing was people who were around in the 70s will know Phaex because it was the first sequenced genome. It also went on to be actually made synthetically for the first time by a group that actually included Clyde Hutchison. But what they observed really relatively early was that Phaex, this is alpha 3, but it's related to Phaex. And what we see there is that the lysis of the bacteria is actually follows a profile very similar to what we see for penicillin or these peptoglycan inhibitors. And that during cell division, you get weakening of the cell wall due to inhibition of peptoglycan biogenesis, and then it pops. Right? It always pops at sort of the septal division point, and in fact, Phaex has a similar process. And so the thought then is that Phaex is just lysine in a similar way. It's somehow engaged in inhibiting peptoglycan biogenesis. So as I said, they worked out all the genes. The genome itself is about 5.4 kb, so it's tiny. We can map all these genes to various functions, and I'm just going to draw your eye to a couple of things here. One is this procapsid gene, D, which surrounds this, and so this is essential for the assembly of the virus. And the other is the gene E, which is the lysis gene. One thing that's really notable about the lysis gene is this lysis gene exists exactly in the same stretch of DNA that encodes for this procapsid gene. And so what that means is that the triplet code that encodes for this D protein, if you shift over one amino acid, you encode a completely separate peptide. And those two peptides have very important functions in the cell. And you can think about this sort of this way. So if I take this green molecule here and I just show one copy of that, that structure is here, but in purple are all of the stretch of the protein that overlaps with the E gene, right? And so it's important to think about this, that any mutations you make in the E gene are also going to make mutations in the D gene, which is kind of cool. All right, so now we know we have this example of this lysis gene. Rai Young basically coined this as the single gene lysis proteins. They are protein antibiotics that target peptoglycan biogenesis. E is here. These other phages are actually much more simpler phages. They're smaller and even less complicated than Phi-X. Of these, we understand 3 of them in their genes. Q beta is a single-stranded RNA phage that inhibits MURA, the first step in peptoglycan biogenesis. The M protein was shown recently, actually encode this gene Lys-M that inhibits this flipase MUR-J. Protein itself contains a single transmembrane domain and this soluble domain attached to it, right? And so this is very tiny 91 amino acid peptide that somehow is able to kill a bacterial cell. If we look at how bacteria lyse, we can see that lysis by a simple T phage looks like this. You add the T phage over time, you suddenly see that they start to die, and then they rapidly all get killed. And that's just because this death gets amplified relatively quickly. It similarly works that way for Phi-X, except it's a slightly slower in terms of its lysis. But what Rai Young's lab was able to show, and this is kind of complicated, I'm just going to follow this here, was that you can get the similar lysis mechanism by just expressing the E gene by itself. You put that E gene in E. coli, and that's all you need to actually get this, get lysis. And so E gene itself is sufficient for lysis. And so in his lab, working Tom Bernhardt and Bill Roof were 2 people working in the group, and they actually demonstrated this kind of clear mechanism for how it works. The E gene with its single transmembrane domain must exist in the bilayer. They identified a protein, a chaperone called slide E, that's somehow involved in this process. And we'll come back to that in a minute. And this is an E. coli protein that has no business being involved in mRNA-Y, but you'll see they interact. The E gene inserts in the membrane and then somehow inhibits mRNA-Y. They could identify mutations in mRNA-Y that were resistant to the E gene, suggesting then that those two would interact with each other, and that was exactly the mechanism that this inhibition would lead to cell lysis. If we then take these mutations identified and additional ones, all of those map to this interface here, which is on the same face where the active site is, but it's on the opposite side of the membrane. All right, so we can actually show that we can do this lysis in our lab, so we can induce this in the presence of slide E. In the absence of slide E, we don't get lysis, and so really slide E is essential, even though we don't think it's not essential for sort of reasons that may have been initially obvious. All right, so here I'm just going to show an alignment. So this is the E gene from Phi-X 174, but across this are just all environmental variants of this. If you purify Phi-X from all over the world, you can see different sequence variants. And the one thing we can see in a picture like this is just all of these sequences align, and where they're similar, this means those are the same residues. And you can see they're completely conserved here. And what's defined as this transmembrane domain, this helix that crosses the bilayer. We see less conservation in this cytoplasmic C-terminal domain, except for some positive charges. We're going to focus on 2 different variants here, Phi-X 174 and ID21. The proteolysomerase chaperone itself has 2 domains, an FKBP domain, which is responsible for catalytically interconverting a proline, and then this chaperone-like domain that's responsible for an insert flap domain. These 2 domains actually are completely independent of each other, even though they're well folded. And this is demonstrated by a solution method where they solve the structure by NMR. And you can see that these 2 domains are basically constantly in motion, suggesting that there's this very high flexibility. Okay, so we basically, maybe I'll just skip over that because it's going a little long. But we basically mapped it out and showed that there's this feature where that proline in the center of the membrane was actually completely essential. And maybe actually, okay. We'll just come back to that. All right, and so what can we do? So Shio Shinaka was the first person to start this project in my lab, and she basically showed that if you express mRNA-Y protein E together, you actually pull out slide E in a 3-protein complex. And that complex, you basically can see with these bands in the gel all represent the fact that we've purified these proteins from an E. coli. We could purify them and then show, so this peak here basically indicates that it's relatively stable, happy protein, and it's purified. And so we can take this protein and try to solve the structure. And so we do this using a method called single particle cryo-EM. And so the idea for this method is that you're going to take your molecule in water, and then you're going to hit it with electrons. And those electrons, just like X-rays at the dentist, are going to actually be penetrant, and they're going to come through your molecules, and you're going to collect a 2-dimensional image of those molecules in that thin ice. And that 2-dimensional image contains 3-dimensional information. And so for each one of these particles, you can pull these particles out, identify where they are, identify how they relate to each other, and then computationally, you can reassemble these to actually build a 3-dimensional molecule. And so that's what we've done. And so the experiment itself involves things like this. This is a cryo-EM microscope at Titan Krios that's actually here at Caltech. This is my student Karen actually loading one of these samples into it. This is one of her pieces of data. And so what you can see here, this is an EM electron microscope image of these yes complex in vitreous ice or in ice. And what you can see, there are these little particles here, which I think you'll all believe show you exactly this atomic resolution detail. In fact, we can zoom in, and what you see here is that, there's three-dimensional atomic resolution information there. You can believe me on that. And so we can take this and computationally solve the structure. And so I'm going to show you that now. So we have our yes complex, right? Yes stands for mRNA-Y, protein E, and slide D. Remember, 2 proteins are from E. coli that never actually interact in the absence of this protein from Phi-X. And then protein E comes together. And so this image that I'm going to show you here, this is a movie. In this movie, this is what we get. So because these membrane proteins have to exist in the bilayer, they're very hydrophobic and they won't be stable in water. We stabilizing them by surrounding them by a mild detergent, just like what we use to wash dishes, although it's much milder than that. And in stabilizing that in a detergent, we can see this molecule, and it can strip away that. And then just to give you an orientation, the periplasm again on top, cytoplasm on the bottom. We're going to strip away the bilayer and now look at this, the structure. And so you can see that, you hopefully see that there are these sort of tubes of densities that are crossing this bilayer that I've indicated here. We can color the molecules in because we know the structures of both mRNA-Y and slide E. And if we do that, we can see our dimer, right? So here in blue is our homodimer of mRNA-Y with active sites on opposite sides. There's 2 copies of slide E that are bound. And then in gold here is our 2 copies of the E protein. And so we can actually look at this at high resolution. And so we actually can see the details. We can build our molecule into this. And so we're going to replace this sort of density model here with an actual molecular model where we've modeled in all the amino acids. So one of the features that's really critical to this is it's completely conserved proline. And if you look at this completely conserved proline, where that occurs, it actually occurs right here where this helix gets broken. Prolines have this property of breaking alpha helices that allows this transmembrane domain to actually go around this helix here. And so in the absence, with that proline there, it's really essential to actually allow that to happen. And we can see that a little bit better here. And so now if we look at how slide E is binding, or how E is binding to mRNA-Y and slide E, the transmembrane domain binds in the groove, blocks the active site, and then slide E sits here on top of this soluble domain here. And so really the mechanism of inhibition is pretty simple. E acts as a wrench that sort of really blocks access here. And so it's just basically a wrench in the works that just basically prevents the enzyme from doing its catalysis. We can zoom on this a little bit here. Here's actually some known inhibitors actually bound to a separate structure of mRNA-Y, but we can dock those into our structure. And what we can see is where E comes in, and it basically blocks the active site. And it does it by doing 2 things. Here we have catalytic residues here and catalytic residues here. It prevents them from being able to come in and do catalysis. But the other thing that it does is it blocks access to the lipid. And so the lipid would come in through the membrane through this groove here, and it basically prevents that lipid substrate from having access. We can also now map all these mutations that we had seen before in mRNA-Y that led to resistance, and we can basically see that they all mapped direct interactions with Phi-X, this Phi-X protein. And so it's sort of interesting. We actually can look at how Phi-X and actually ID21, we can actually solve structures of both variants. And so Karen and Evelyn went on to solve a structure of this ID20 of Phi-X itself. And I won't go into the details here just because of the sort of time, but what we learned from this basically was that they essentially, as expected, look the same way, but there's some sequence differences that actually result in function. And we can go back and actually test those mutations directly, that these mutations, where we see things like this completely conserved, where we see this completely conserved arginine is, or completely conserved lysine, sorry. So there's this residue, this lysine that's positively charged that interacts with these negatively charged residues. And just to sort of draw you to this lysine here is, if we make the mutation in the lysine, so here, just to follow this experiment, in the wild type case, we get lysis in orange. If we make that mutation, we no longer see lysis. And so we've now, using the structure, identified residues that were critical to this function. We also can map out things like sort of how Slide E interacts with this and sort of the sort of novel features, this unexpected evolutionary quirk of bringing Slide E into the mix here. One thing I didn't point out is that the E proteins cross over, and so a single E gene actually interacts with this one Slide E IF domain here, and then the other Slide E IF domain here. So they cross over here in the middle of the molecule. And they do it in a way that's sort of interesting and actually reminiscent of structures that have been seen, peptides bound to this before. And so if you just sort of look at this interaction here, sort of oriented in the same way, basically see that this groove is occupied by sort of an extended peptide, as we see from 1E. And then there's a surface here, hydrophobic surface, which is docking onto our helix, which is also where a peptide binds in this case. And then the rest of the molecule would be going out, and there'd be a proline binding in this prolyl isomerase domain. All right, so conclusions. So I hope you told you a story that you at least found intriguing. We certainly have found this whole experience quite thrilling. We know it's actually a very simple mechanism for how this works. Protein E is mRNA by basically blocking access to the active site. Slide E doesn't appear to actually take advantage of its sort of normal role in the cell to act as a chaperone to help proteins fold. Instead, it just it binds to this complex and stabilizes protein E, so that protein E can inhibit mRNA. There's that completely conserved proline that allows this kink here, and that seems to in no way interact with Slide E. And so it seems that really that isomerization role for this chaperone isn't really critical, which was unexpected. I didn't have time to show you this today, but we do believe there's likely a very important role for lipids in this function, and we have some insights into thinking about that. And what's cool is this actually is just the tip of the iceberg for the way that phages have actually taken advantage of making these small peptides to kill bacteria. And so this is the start. We're really interested in sort of expanding the way of thinking about this and leveraging it for development of new tools. So what are the open questions? We still don't really understand the mechanism of mRNA, and that's something my group is actively interested in. One sort of interesting question is, how do you evolve something like this? And so I have some simple insights into that, but it'll take some time to kind of work through them. And then obviously the real key is, can we use this to engineer phage? Can we use it to think about leveraging this interaction to develop novel drugs? And this is an area of excitement for our group. All right, so finally, I just say, this is sort of the classic pandemic picture, needs to be updated still, but of a group meeting. But really, this is really a team effort by a lot of really great people, and I've hopefully mentioned all of them along the way of the folks who've done this work. But this is funded by a number of different sources, and it's really been a fun project. And it's actually the synthetic chemist is Mitchell Carosi of the laboratory in Tennessee, who I didn't have a chance to talk about his work. But I do want to thank all of these folks, and I'll be happy then to take any questions.
Thank you so much, Dr. Clemens. Oh my goodness, the Q&A is blowing up. So I'm going to try to start from small to large on this. Just a story, and I think the history of science, and science is so relatable. David Lewin writes, while I was a biology undergrad at Caltech, I heard the story about how Bob Sinsheimer obtained the Phi-X phage. He had written to a researcher who was working on the phage, who replied with a letter that he would not provide Sinsheimer with a sample. However, Sinsheimer was able to isolate the phage from the letter and went from there. So
yeah, no, it makes sense. It would be everywhere. Yeah.
Okay, so and I think again, starting from the small, more technical, Dave Zobel writes, Biomolecules get much of their power from their 3D structure, as your illustrations and videos clearly show. Beautifully done. Yet students rarely learn about how utterly structure informs biochemistry until their college years. Instead, we drag them through mathematics, electrodynamics, and other abstractions that are hard for young people to visualize. If a picture is worth 1,000 words, why aren't we teaching basic mechanisms of biochemistry to younger students? Any thoughts on that?
Yeah, well, it's interesting. It's an interesting point. I agree with you. I think the beauty of structure is it really does make things very simple often to understand how things work. And so this, as you say, a picture is worth 1,000 words. So a three-dimensional picture, I don't know how much that is worth, but it's worth a lot. And so, you know, I think there's some merit to it. I think there's some to teaching it at a younger age. It's certainly seeing my first three-dimensional structure. Actually, I was probably a junior in college before I kind of really grasped what three-dimensional structure was, and it blew my mind. It set me on the path that I was on, that I've been on ever since. And so I do think it's realistic to say that if we can engage students at an earlier age, you know, that would be transformational. Because one of the things, you know, and this is, I think, what made Caltech great, especially in the 50s and 60s, still today, but I think it's this sort of merging of this sort of gradient of sort of physics and biology and chemistry and everybody being in the sort of same environment to allow us to think about life, you know, on many different levels. And, you know, structural biology really is that tool. But I do think, you know, it's sort of hard to capture it without knowing it's not hard to capture understanding without knowing a lot of other parts that go into it. And so it'd be interesting to think about how you could teach students to do it. Although, to be fair, you know, lots of high school students have come into the lab and done structural work. So it's not that you can't do it at that age.
And lensing a little bit outward, there's a lot of questions in this category, but Brian von Hertzen, PhD, how far away are we from a new class of antibiotics based on this research?
Yeah, pretty far if you're asking for small molecules. I think we do have lots of ways that we've thought about new mechanisms that you can imagine for development of small molecules, and I think we're going to pursue those. But that's, you know, decade-long path. The bigger question is, you know, can we leverage this to understand phages and for phage design and think about phage therapies? You know, my guess is that phage therapy is going to overtake small molecules in this regime probably faster. And the reasons for this are actually multifold. One is that the pharmaceutical industry has zero interest in small molecule antibiotics for bacteria. And you think, well, this is a huge problem. Why wouldn't they care? And the problem is there's just no money in it. So, you know, that's on two levels. The first level is if I solve this, if I get a new type of a small molecule antibiotic that works really well, I cure you, and then, you know, it costs me $10 to make it. Maybe I charge you $50 for it. You're cured in a week, and then now I don't treat you with it anymore. And if I'm really good at it, I cure everybody, and then there's no continuing money in it. So there's sort of that dollar, corporate way of thinking about it. The other problem is, and this is probably the more relevant one in terms of why nobody will do it, is because if I develop a new small molecule antibiotic, I talk about, I mentioned the fact that vancomycin, minomycin are these antibiotics of last resort. New antibiotic drugs are going to be put on the shelf and never given to anyone unless they have a multi-drug resistant bacterial infection, right? And so if you take the economics of sort of, treating, only treating maybe the one percentile of patients who have bacterial infections, the numbers get even worse, right? And the reason you don't want to do that is you don't want to develop resistance, and so you don't want to treat people with a new drug unless they need it. And so it's a sort of complexity of how you're going to market a new molecule and the way you're going to profit off of it, which I think has pushed that realm back onto the academics as the place where we're going to discover these new small molecules. But I do think because it becomes a sort of small market problem, phage therapy is one of these things that actually can really handle things on that level. The problem with phage therapy as it stands right now, because we don't have sort of designer engineered phages, which may come, and some of this research may actually help with that. But the problem with phage research is the way that it works now. You culture a phage, and so in this case, for that case I was talking about, this Acetobacter baumannii, they basically cultured it, grew it up, then you throw in a bunch of phage, and if those phage can kill it, they reproduce themselves. You take those new phage, and then you just you farm the phage on the bacteria that you're trying to kill. And that works really effectively, but it's a process that's very patient specific. And so you would have to then imagine that every lab would have to have some, phage lab there to be able to treat people who weren't responding to antibiotics, which is doable. And I think it will be very doable, but it is a whole different way of approaching medicine for that.
And would it be, and perhaps this is a simple but fascinating question, would it be a pill, injection, a liquid? How would the phage be?
Well, in that particular case, they gave it to the patient intravenously. So, but you could imagine, it just depends on where the infection is, right? So a topical infection, you'd probably just, wash over the wound, and that would be enough. But, if it's an internal infection, phage aren't going to travel from the gut to the bloodstream. And so if you have to get into the bloodstream, it would have to be an injection. But, that's all completely doable. And this is true for, vancomycin and minomycin are pretty awful drugs to have to take. And then they also are intravenous as well. And so, if you have one of these infections, you're going to be in a hospital with a drip with something in it.
Daniel Dobkin notes, amazing presentation to allow a physics person with very little biochemical background to follow at least part of this complex story. Thank you so much. That's a shout out from your colleagues in physics. To slightly larger questions, if you may, Eduardo Kutulink from Monterey, California. This is for you. Is the prediction of superbugs surpassing cancer as the cause of death still by 2050 still a strong possibility?
I don't want to predict pandemics, right? But I think we realize now that when microbiologists issue threats, they're not idle threats. And so I don't know if superbugs, superbugs are going to make things complicated, right? And so, whether or not, I think they're not going to be airborne, right? And so it's not like you have to worry about getting these infections, in the same way we do viruses. But the reality is contaminated food sources, livestock and things like this. Our whole food chains could get completely contaminated by some of these, and that could be pretty devastating. And so you can imagine that could be a major problem, certainly in parts of the world where they don't have, necessarily clean water or access to sort of, sanitation resources. This will become a major problem. It's hard to imagine that bacteria will kill all of us, but I do think that that bacterial infections, it's fair to say that people on this call are going to encounter this in their lives, and so either themselves or family members, and so multi-drug resistance is going to continue to be a major problem. What happened in this particular case in Egypt, it was just a spoiled dish apparently that he ate. Everybody ate it, but for whatever reason, this one bacteria took off in him, and that was it, right? And so he if this hadn't happened, he literally probably would have died a week later. Phage saved him, but that was, it's not like he was doing anything risky. Let's put it that way.
Right, and to that, are there environmental concerns with using phage? The bacteria will build resistance to them as well.
Yeah, and so that's I think both the strength and the weakness of any of these treatments. The problem with small molecules is you have to completely design a new small molecule every time. There's some resistance almost always. The beauty of phage is in this battle too. And so, what you would typically do in a phage cocktail is you don't just give one phage, you give 3 or 4 different classes of phages or types of phages that all target the same bacteria in your cocktail. And so the likelihood that your infection is going to be able to survive this onslaught from all these phage who are fighting to kill you is very small. And even so you don't expect to kill every single bacteria, but your immune system's working too, right? And so I think the whole point is antibiotics basically to knock down these bacteria to a point at which your immune system can deal with it. But that's the trick. And the thing is that because you can do this on an infection by infection basis, you don't necessarily, you're not going to have this sort of problem of resistance in the environment that you're going to be dealing with. And even if you do, the beauty is that the phage wants to win too, right? And so you're basically, it's like an attack dog versus, having a sword, right? Like at some point, both are effective weapons, but an attack dog is going to keep fighting even when when you drop your sword, and it's kind of the same way. Poor analogy, probably.
Sort of an interesting history question. If you'll speak to it, did Phage therapy's popularity suffer because the research and knowledge was done outside of the major Western science centers in the 20th century.
Well, it's certainly true that in the West, they, and even here at Caltech, people thought about this a bit and worked on it a little bit. But I think a lot of great research was coming out of the Soviet Union on this area, and it was really ignored by the West and was not treated with a lot of respect. I think that's probably fair to say. It's hard, right? I think, treating people, it's going to require a new way of doing medicine to sort of think about this, but we can do it. I mean, the tools are already in place, and it's just, and in fact, the regulatory bodies used to be, a decade ago, really fought against phage as a therapy because it's just really hard to regulate something that's constantly mutating. But they've come around, and so I think the reality is that these are going to be the way that people are going to be treated. Some percentage of the population is going to be treated pretty regularly with this. And it's certainly, if a family member who had an infection, I would go down to San Diego and talk to Dr. Strathy and her team and start there.
Well, thank you so much for this fascinating talk. Thank you, Dr. Clemens, and we'll look forward to more research to be continuing. Thank you so very much.
Thank you. Thank you all for listening.